2009. He has been involved in biotech in many different companies for a very long time, a very successful career. And he holds a BS in biochemistry from College of Charleston, South Carolina, the PhD in pharmaceutical sciences from the Medical University of South Carolina. By the way, great state, a beautiful state.
Yeah, that's why I stayed here for a while.
And has an MBA from Carnegie Mellon. So he will talk to us again about his work and his involvement in moving nanotechnology and partnering with academia all the way to the bedside. So thank you so much.
Zahi, thank you very much for having me here today. It's a pleasure, and I have the daunting task of keep you guys entertained right after lunch when everybody's ready to fall asleep. So I will do my best to hopefully make this somewhat interesting. So the focus of this talk is less about features and functions of nanoparticles. I think you guys know a lot about that. What I've tried to do here is to tell you what it takes to develop successful drugs, and to give you some insights in terms of where we stand in cerulean at this point. So we're in the middle of phase 2 development. So we're clearly not at the goalpost yet. We don't have an approved drug yet, and there's still years and years of work ahead. But we're certainly well beyond animal models and early clinical testing. So I want to just give you a perspective of how that works. And I also hope that you have some questions. So I'll try to keep it relatively short with the presentation and then be available for questions. So the way I wanted to organize this, to just give you an overview, what do we do, talk a little bit about the promise of nanopharmaceuticals. That's probably nothing new to you, but still makes some sense to talk about a little bit, and then showcase our lead compound, CLX-101, of what we actually see in the clinic and how we got there. So when you look at the arduous history of the fight against cancer, it's actually somewhat shocking when you look at 1970 was the year in which President Nixon declared the war on cancer. In 1970, the 5-year survival rate across all cancers was about 50%. Today, so more than 40 years later, and I don't know how many billions of dollars have been spent since then on cancer, that survival rate is still below 70%. Now, in some cancer types, we have made fantastic progress. We have literally eradicated the disease. You can look at other cancer types, and unfortunately many of them, non-small cell lung cancer, ovarian cancer, brain cancers, I mean, you name them, we still have virtually nothing. And so when you look at that, you can almost look at it as a book, chapter after chapter after chapter, and each of these chapters have certainly made very meaningful contributions on the journey of tackling cancer. And we believe, we call this somewhat colloquially smart bombs. Smart bombs for the reason that we target into the tumors very toxic payloads. You've heard the talk from Jeff earlier from Bind, excellent progress of what Bind has done. And we believe that this whole notion of taking either antibody drug conjugates or nanoparticles can be the next chapter and can get us actually at least closer to managing the disease, not necessarily a cure yet, but get us down this path where we need to be going to make a really meaningful difference to patient lives. Very, very simple view of the world. What you see here on the left-hand side is just a cartoon of normal tissue and tumor tissue on the right-hand side in red. We all know that the pore size of the vasculature in tumors is larger, so that's not a surprise. So you can take nanoparticles, if you design them properly, they will slip through these leaky vasculature pores into the tumor. And then the drug can be released. So far, the concept, nothing really new and exciting about that part. I think that's been pretty well established. What we do is we make these nanoparticles, and then on the inside, we have the active drug covalently bound through a linker to the nanoparticle. We call the whole thing dynamic targeting. Why dynamic? For a couple of reasons. The first one is this leaky vasculature uptake. Again, if you design a nanoparticle well, you get leaky vasculature uptake. The second step is then what happens to our nanoparticles is actually uptake into the tumor cells through macropinocytosis, which is overexpressed and upregulated in tumors. So that actually gets you from the tissue in the tumor into the tumor cells. And then we have gradual release of the payload, the drug, from within the tumor cells. So that's the third step. Now that last step may very well be the most important step, particularly when you have drugs where exposure drives activity. Clearly, if you have drugs where high concentrations are driving activity, you don't want to slowly release them. But in cases where slow release and exposure is important, that's exactly what you want. So I mentioned before, this is a somewhat time-consuming process. It's not something quick. So when you look at the history of the company was founded in 2006 based on MIT IP from Ramses Sekaran's lab, and that gave the foundation of the company. We then in 2009 brought in complementary IP from Caltech, Mark Davis's lab. So Mark Davis was actually also supposed to be here, is unfortunately not here today. So I'll take a lot of the work that he started. CLX 101 actually originated in his lab. And we brought it in 2009. So I'm actually cheating by saying this all started here. You don't just drop into phase one, right? There's years and years of work beforehand. So Mark Davis and his team deserve a tremendous amount of credit for years of work beforehand that led to us being able to bring it in and then do the phase one and get into phase two development. And then you can just see, without going into all the details, there's a lot of stuff going on now in terms of different tumor types that we're currently pursuing. Some more as drilling test holes where we think this could be interesting, others with a clearer focus of we say we know it's interesting. So the next product, by the way, is based also on the platform, and we expect that to go into the clinic next year. Now, in order to do this, you need to actually have a broad team, and on the other hand, you also need to have a strong investor syndicate. It takes both. So while on one hand, you clearly need the scientific expertise to get you to the clinic, but you then actually need to start thinking about how do you build a company. So it really doesn't matter what the details of the people are here. I just wanted to use it as an illustration that this becomes very cross-functional. And at the same time, you're constantly, if you're a guy in my shoes, you're constantly chasing money because drug development becomes very expensive once you're in the clinic. The typical cost per cancer patient that you do in your study is north of $50,000 per patient. So if you want to run a 100-patient trial, you can do the math pretty quickly in terms of how many $1 million you need to be spending to run effectively one experiment. So what is the promise of nanopharmaceuticals? Very, very simply put, and again for this audience I'm grossly oversimplifying it, but sometimes being simple can be helpful. When you look at conventional therapy and you look at your target tissue shown in solid lines here and you're looking at your systemic drug exposure, you're actually getting from many oncology drugs the exact opposite of what you want. You have high drug levels in the distant tissues, and you're lucky if you're getting as much into the tumor as you would like to have. Simple efflux pump upregulation contributes to drugs just getting shuttled out of tumor tissue. So you have the perfect combination of what you don't want. Lots of drug where you don't need it, causing side effects, and not enough drug where you do need it in the tumor. So if you could change that into achieving actually higher concentrations in the target tissue in the tumor and having relatively low systemic distribution, that would be ideal. So where are we on that? Let me give you a very quick snapshot of what our lead molecule actually is. It's a simple backbone polymer here with a linker where you attach the drug camptothecin, and it self-assembles into these nanoparticles about 20 to 30 nanometer in diameter. While there is a somewhat heated debate, is 100 nanometer better than 80 nanometer, better than 20 nanometers? Who knows? I think there's so many factors that contribute to it, but we all know it needs to be small enough that it can slip into the tumors and large enough that it doesn't slip out through the kidney and gets very rapidly renally eliminated. And the lower bound for that is probably around 15 nanometers or so. So what we have here is an ultra-potent topoisomerase I inhibitor, and actually an old compound. It's a natural product, so you can compare that to Taxol, which is a natural product, was discovered to be extremely potent. Same thing here with camptothecin. However, camptothecin was so toxic that when people originally tried to develop it, it was impossible. Patients had horrible side effects and the development had to be stopped. So we are now taking an ultra-potent inhibitor of topoisomerase I, and by putting it into the nanoparticle here, we effectively mask the toxicities and get it where it needs to go. And we can dial in the speed at which the drug comes out by using different linkers. So that linker technology allows you to actually get the release kinetics that you need for optimal activity. Keep that in the back of your mind because it's going to become important as we go further in the story. So you saw the cartoon earlier in terms of the preferential tissue uptake. What you see here are a few slides. The first one is from an animal where you can actually see the nanoparticles here. This is a triplet enlarged in a tumor in a xenograft model here. And you can clearly see it. It's actually in the tumors itself where you want it to be. Animals is nice. That's good. That's where you start. Here's a triple-negative breast cancer patient. And so this is a lady who had a biopsy close to her, a tumor close to her chest wall, so it was easily biopsyable. And so when she came back, our drug is given every 2 weeks. So she got her first dose, and then 14 days later came back for her second dose. A biopsy was taken before she got her second dose. And camptothecin, it's a heterocycle, so at the right wavelengths it actually fluoresces. So you can see here, excuse me, all these green dots. Blue are the nuclei of the cancer cells. You can see all these green dots in the tumor here 2 weeks after a single dose of the drug. So it's pretty exciting because you know even 2 weeks after a single dose, there's still drug there where you want it to be. Now the big question that we've always been hunting after was, do we really have differential uptake versus healthy tissue in humans? We know we have it in animals, but a lot of skeptics are saying, well, humans are different, this whole leaky vasculature stuff, I don't really know if I buy it, and you've probably heard the arguments. So we were very fortunate here that in a gastric cancer patient, we did a biopsy 48 hours after therapy. And again, you can see here in the tumor, these green dots, again, the camptothecin in the tumor. And this was done in Mark Davis's lab, by the way. We continue to collaborate with him. And then the biopsy also included some healthy tissue right next to the tumor. So we had, in fact, the ability now to look at healthy tissue and tumor tissue from the exact same patient at the same time point. And you can see here, you're only in background noise. There is no camptothecin that you can see. So that was the first time that we were able, actually, in a human being, in a patient, to show that there is differential uptake, even 2 days after giving a dose of the drug. This one is just a beautiful picture where you can, in an animal model here, in an ovarian model, you can see this blue haze. That's the camptothecin, the green particles, CLX-101. You can see the nuclei of the cells. So you can very clearly see the drug is right next to the nuclei where it needs to be in order to do its job. When you then look at tissue distribution studies, you can see what we've done here is compare camptothecin, the actual payload on the right-hand side, with CLX-101 on the left-hand side, but we're measuring only the free drug. So this is not total drug in the nanoparticle, this is the free drug that's actually released. And what you can very clearly see here is while you have very little in the tumor and most of it in the liver, when you look at camptothecin, you get massive increases. So your ratio changes completely, excuse me. Your ratio changes completely between tumor uptake and rest of body uptake when you use this technology. You can run animal model after animal model, and in each case you see that the drug in itself is very active, but here a couple of synergy studies. So you can see here paclitaxel in this particular model, even though it is active in ovarian cancer, very little incremental activity over placebo. Our drug at a relatively low dose, nice activity, but when you combine the two, you get really beautiful synergy. So you can do that. You can do that with radiation, and I'll explain to you in a few minutes why we have this synergy here. And again, you see radiation by itself, very little effect. 101 at a very low dose, little effect. You combine the two, you get synergy. So, and then there's reams and reams of data where 101 in different tumor types outperforms the relevant comparators here. So we can run these models up and down, which gave us a lot of comfort. Yes, we have something here. This is really interesting. We need to try this in humans. So how has it been going? What do we know today about the agent in the clinic? Let me start with what may at first appear a detour. I don't know how many of you have heard of Hit1 Alpha, hypoxia-inducible factor 1 alpha. Hit1 alpha is regulated by the oxygen gradient in our tissue, under low oxygen conditions, HIF1 alpha gets upregulated. And when HIF1 alpha gets upregulated, it becomes a master regulator for a whole bunch of downstream pathways here. And so this is actually from a Nature Review article that was published last year. And it's been fantastic to see how some of these disjointed observations about how tumors survive have now all been linked back to HIF1 alpha. So HIF1 alpha has emerged as an undruggable master regulator of cancer survival pathways. So a lot of the reasons why cancer patients die are not the primary tumors, they're actually the metastases and the dedifferentiated cells that actually take on new genetic patterns that make them harder to treat with monotherapies. So wouldn't it be nice if you could actually hit this target here somehow? Well, it turns out that when you look back at work that Giovanni Melillo at the NIH did, he did an empiric screening of the compound database at the NIH, and he found the camptothecans, and there are two approved camptothecans, irinotecan and topotecan, still very, very toxic drugs, very poorly tolerated. They hit HIF1 alpha, but not in a durable fashion. So you have very short inhibition of HIF1 alpha. So purely empiric, it seems to be something about the camptothecans that make them good inhibitors of HIF1 alpha, but not durable inhibitors. So that got us interested, because we said, well, if we have a slowly releasing camptothecan that goes into the tumor, we should be able to inhibit HIF1 alpha. And you can just see here across different tumor types, yes, we actually do inhibit HIF1 alpha. So that was pretty interesting. We then did just a simple experiment, a low dose of 101 given, and then a time course measurement of how much HIF1 alpha suppression does one get. So I said earlier, without the nanoparticle slow release, you wouldn't get some of the properties that we're seeing here. You would not get a 1-week suppression of HIF1 alpha if our drug wasn't slowly releasing, because we know HIF1 alpha gets constantly resynthesized. So we know we have a durable HIF inhibitor. We know that HIF seems to be pretty important in some of the downstream cancer pathways. Now, what do we do with that? Right? How does that actually play out? Well, here's an illustration of how the biology can guide the drug development of your agent. So when you think about just some examples of approved drugs, first one, Avastin, is a Roche Genentech product. It currently sells $7 billion worth of sales per year. It's the largest oncology drug by sales. Zaltrap is a new Sanofi product, Votrient, Nexavar. All the major players have some compound that effectively inhibits angiogenesis. So these are anti-angiogenic agents. But what do? They constrict blood flow. Right? That's pretty much what they do. They constrict blood flow, which drives up hypoxia. So you get low oxygen in your tumor, at which point you upregulate HIF. And I showed you before that these downstream pathways are all regulated by HIF. So in a perverse way, by giving the $7 billion a year drug Avastin, you actually create downstream problems. And lo and behold, when you look at what we've learned about Avastin over the last few years is, and most notably, the FDA actually did something unprecedented a couple of years ago. They revoked an indication in breast cancer for Avastin. Why was that? Well, what you see with Avastin is beautiful shrinkage of tumors. So on the CT scan, you guys can probably make beautiful pictures at every which way till Sunday after you give a VEGF inhibitor, because what they're fantastic at, they shrink the tumors. Right? So you have this beautiful shrinkage of tumors, you get responses, it looks very encouraging. But then often you don't have a translation into an overall survival benefit. So these patients have temporary remissions and then they bounce back, and they actually bounce often back with more metastatic disease. And we really didn't understand until the last few years. Why is that? What is going on here? And so now we increasingly have HIF1 alpha implicated as the bad actor here, which makes it even more important to saying, if you could interrupt that loop, that would be pretty cool. So what we've done then is to say, well, if all of that is true, can we prove synergy between our drug and some of these VEGF inhibitors? And so on the slides here, what you see in the top is again HIF1 alpha levels as a percentage of control. When we give our drug in blue, I've already shown you the knockdown, you see suppression of HIF. In purple, you see each one of these representative approved drugs. They upregulate HIF, so you can see a very clear upregulation effect. And what was really exciting for us is when you then combine the two drugs, so in this case 101 plus Avastin, or 101 plus Zaltrap, or 101 plus Votrient, you see this HIF suppression again. So it means even in the presence of one of those bad actors, you get HIF suppression. Now, does that matter? Well, when you look here at the survival plots of the animals, you can see again Avastin alone, our drug alone at a low concentration, and then the same two drugs combined. Yes, you see a very distinct benefit here by doing these combinations. The other thing that I mentioned to you was cancer stem cell influence. So here's work that was done at the University of Michigan in Max Richa's lab. He has a model in which he just simply grows up the tumors for 35 days, and then he exposes them to drugs that he wants to test. So in this case, he used our drug in blue, Avastin in purple, and then the combination again in green. And the first model is actually mostly an incubator model. So what you're doing here is you're not really that interested what happens. What you want to do is you want to take 100 tumor cells from these tumors of these pre-treated animals, and you want to then implant them into the next animal. And the idea here being, if you have lots of cancer stem cells, they will take pretty readily, because now you have dedifferentiated cells, they're easy to establish, they will grow. If on the other hand you have fully differentiated cancer cells, it's actually quite hard to grow a new tumor from 100 cells. And so what you see here is, again, with Avastin, the beautiful evidence that Avastin alone actually drives up cancer stem cell formation and drives up secondary tumor formation, so effectively a model of metastasis. Whereas with 101 you have very poor take, and then again, when you combine the two, it completely obliterates the effect of Avastin. So that got us really excited of saying, wow, this all seems to fit together very nicely. We're in a position where we can now hit Hit1 alpha, while drugs like Avastin cause some real problems for patients. All of that really doesn't matter all that much if you have a drug that you can combine. And so what you're seeing here is the safety profile of our drug, and what we're doing is we're just plotting the top 10 adverse events that are seen in patients, and we're grading them, we're color coding them by severity. So grade 3, grade 4, which is the ones that you really worry about as a clinician, are in red. Grade 1, grade 2 in oncology is something that people don't worry too much about. And so you can see the two approved camptothecin analogs on nasty drugs. I mean, you can just look at it graphically. I mean, the number of grade 3, grade 4 toxicities are profound. So they have a short half-life, which means they don't give you durable HIF inhibition, and they're toxic as can be, which means you can't really combine them. On the other hand, we get durable HIF inhibition, and it looks like our safety profile is quite favorable for combination. Then on the right-hand side, just to give you a comparison, Gleevec, Novartis' drug for GIST, is actually one of the first chronic therapies for cancer. Patients take it for years just to know this is what a safe drug looks like, just to put that in perspective from a safety profile. So where does all of that lead us to? It leads us to saying, well, if we want to optimally develop our drug, we know that it's a topoisomerase 1 inhibitor. All the camptothecins do that. It's validated through irinotecan and topotecan. So that part we knew all along. And so we thought by tumor targeting we're going to have a better and safer topo1 inhibitor. What we have learned is that we actually inhibit Hit1 alpha because of the properties of our nanoparticle and the slow release in tumors. We're now bringing Hit1 alpha, a previously undruggable target, into the mix. And we also know that we have a safe drug, so therefore it should be combinable. Well, what's the obvious drug for us to combine with? Avastin. Right? It's a very obvious choice for us. Our drug is given IV every 2 weeks. Avastin is given IV every 2 weeks. I've shown you some of the data that suggests we have very strong synergy between the two. So this became then a question of saying, well, can we show some of that in the clinic? So we're currently on a path that starts to really maximize that topo1, HIF1 synergy and combinability. While you always have to show in monotherapy first that you have activity as a springboard before you can go into combinations, we believe the real prize is in combination therapies. So I'll just give you a couple of snapshots here. So we just finished enrollment of a phase 2 trial up in Boston with the Harvard Teaching Hospitals, where we're taking relapsed ovarian cancer patients. So these are very, very sick ladies. They have very few treatment choices at this point. And the idea here was not to say we're going to go into monotherapy in ovarian cancer. The idea was we know HIF is overexpressed, so this is a HIF overexpressing tumor type. We have very strong preclinical data. Topotecan is an approved drug, so we know that that kind of compound class works in this indication. And there's a big unmet medical need. Well, what we have is already, and we don't have all patients finished yet, but we already see in monotherapy that we have RECIST responses, very nice RECIST responses in these patients. In parallel, we also started a trial in combination with Avastin. So that was my last slide where I said to you, Avastin is kind of the logical combination agent for us. Now we needed to prove it, so what better indication than renal cell carcinoma, because this is also HIF overexpressing tumor type. And Avastin is an approved drug here. So here it's very easy for us as a small company to say we just simply take the current standard and we add on top of it and see where we go. So here, very exciting that we can actually generate some data that, unfortunately, probably the most important, you can't see the most important part of the slide, which is there's a light gray for each of the prior drugs, the number of months that these patients were on therapy. So let me walk you through that real quick. So we have, at this point, only 10 patients on the trial. It's an actively enrolling trial, but we're already seeing 3 RECIST responses out of these 10 patients. So let me put that in context. These patients are heavily pretreated, so you can see the prior lines of therapy here. In these prior lines of therapy, the median duration, the gray bars that you can't see, was 2 months for these patients. So on average, these patients had about 2 months for each of these agents. They then progress through, and nothing really touches them. You have them for 2 months on a drug, sometimes for 3 months, sometimes for 1 month, but very short treatments here. And then you give them our combination of 101 and Avastin, and you end up with, even in 4th line, you see these very durable responses, patients doing well, RECIST responses. And when you go into the literature, for these patients, you normally would expect a 2 to 4% response rate. So 1 out of 50, 1 out of 25 patients you would expect to actually get a response. So to see 3 out of 10 seems to fully confirm that this synergy between Avastin and 101 is real. So that sets us up to now go into combination therapies, because we know the drug is well combinable. We've just generated this data with Avastin. We've had no adverse events that limit the combination between 101 and Avastin. We know we have a HIF inhibitor. So we can now go into a combination trial in ovarian cancer, where we can build on the success from the monotherapy trial and continue enrollment in combination with Avastin. So that trial is just starting up, literally getting going at Harvard right now. We will then also go into neoadjuvant rectal cancer, which is another tumor type that's very HIF sensitive, upregulated, where our drug can have some real benefits. Let me just spend a minute on neoadjuvant rectal cancer, just to give you a clinical perspective of what we are aiming for here. When you think of a typical rectal cancer patient, they're fortunate that upon diagnosis, most of them do not have distant metastases yet. So for those patients that don't have distant metastases yet, again in rectal cancer, fortunately the majority, the typical therapy is 6 weeks of radiation and 5-fluorouracil capecitabine in combination, followed by a break, roughly 6 weeks, and then surgery. Some patients are lucky. At the time when they have the surgery, the tumors have actually been completely debulked, and the surgeon really has nothing left to take out. There is no visible tumor. And when the pathologist looks at the tissue and finds no tumor cells, it's called a pathological complete response, or PCR. Currently, we're achieving about 15 to 20% PCRs. That means about 80 to 85% of the patients are not cured. However, the 15 to 20% of the patients are effectively cured. 5 years out, they're still doing great. And this is one of the few cancer types where you can say, yeah, at least 15 to 20% of the patients you can cure. But we also know that there's, of course, lots of headroom here. It would be nice if you could cure more than 15 to 20%. So the radiation therapy in itself causes hypoxia because it destroys cells and it destroys blood vessels. So you create a hypoxic environment, which again, it upregulates HIF, and it then drives metastases, it drives dissemination of cells. So you'd want a HIF inhibitor in that context. Now, another effect of radiation is, and the desirable effect, is it causes single-strand breaks of the DNA. But the cancer cells then repair these single-strand breaks often, making radiation less effective. The ideal drug would be a topo-1 inhibitor, because topoisomerase 1 religates single-strand breaks. So if you were able to give a topo-1 inhibitor in combination with radiation, that should be highly synergistic. So for those reasons, people have actually tried to give irinotecan, the approved camptothecin analog, in combination with radiation and capecitabine, and that does seem to move the pathological complete response rate up. So it's better than what we currently see. But unfortunately, the side effects of irinotecan are GI toxicity, and if you're a rectal cancer patient and you get already irradiated and you have 5-fluorouracil on board, you don't want additional GI toxicities. So it's a proof of concept that a camptothecin can help you achieve higher PCR rates, but it's also at the same time a limitation, has not been adopted clinically for the toxicity reasons. So it's a beautiful opportunity for us, again, take a big step back and say, well, what do? We have a tumor-targeted camptothecin-containing nanoparticle that's well combinable, that inhibits Hit1 alpha, so it helps with the whole hypoxia angle here and the metastasis angle. And it's a topoisomerase 1 inhibitor, so it's a radiation sensitizer. So that should allow us in this trial to achieve higher pathological complete responses. That's the goal here. And it is also really exciting in that you can actually measure that in a short time period. Often in cancer trials you have to wait for a long time, particularly when you're trying to achieve cures or close to cures. But pathological complete response is measurable in 3 months, and it's directly correlated with survival. So that's another exciting opportunity for us. So I'll wrap it up by saying, well, maybe provocatively here, I don't know how many of you remember the discovery of HIV and the recognition that here we have a new killer. A drug was not on the horizon at first, so in the early 80s, people didn't know much about AZT yet. All we knew is that patients were dying, and were dying of this viral disease. Then AZT was discovered, but it was a horrible drug, toxic as hell, and it didn't really do much for patients. It gave them a couple of extra months. Then the first protease inhibitors came along, and so people were at least able to switch. After they broke through with AZT, they got a protease inhibitor. And then people started to think differently and said, well, rather than sequentially treating a virus that mutates very rapidly and just running very quickly out of options, what if we could combine? And that really started to usher in a completely new chapter. So the first combinations were 2 drugs that helped some, but also didn't last for that long. And it was really with the advent of the first triple therapies, where you were able to shut down multiple pathways in the virus at once, that made it impossible for the mutations to get out from underneath the drugs. And we're now at a point where we have these quads, where you have 4 different mechanisms, 4 different pathways shut down at the same time. And when you just step back now and say, wow, in the 1980s we had nothing. In the 1990s we had some crappy drugs that were the starting point. And here we are today, and we can effectively treat HIV patients that they have a normal life expectancy. And that's just absolutely remarkable. And the only reason why that was possible is because people started to combine drugs, and they made them safer so that they're combinable in the first place. Now, my prior job was head of R&D and corporate development at Cubist. It's an anti-infectious company, and so maybe I'm somewhat biased, therefore, to bring some of the anti-infectious parallels into the oncology world. But I believe we have something to learn here. So rather than trying to have sequential therapies in oncology where we know resistance happens, for the same reasons as in virology, we need to move more into the direction of combinable agents shutting down multiple pathways at once, which is why I give you the example of CLX-101, where we're shutting down Hit1 Alpha, topoisomerase 1, we're combining it with a VEGF inhibitor. So we have 3 pathways. It's probably still not going to be enough, but it's going to be a step in the right direction. And most importantly, we're doing it with high quality of life. I think that's the other thing that we need to keep in mind, that if the therapy for a cancer patient is as bad or worse than the disease itself, we're really not doing a whole lot of good for the patients. If on the other hand you can have a drug that has a nice safety profile and you can combine it and you provide quality of life and hopefully longer life, then that's something that's worthwhile working pretty hard for. So I'll leave it at that. Any questions that you have, I'd be happy to answer. Thank you.
So excellent. I mean, we've seen different strategy to address a very important topic here. So anybody has some questions? Maybe I ask. Oh, there's one I didn't see. Oh, François. Yeah.
Yes, arenatican have shown to be synergistic with TRAIL. Have you ever tried to use
Synergistic with what?
with TRAIL or any anti-DR5 antibodies? Have you ever think about putting both together?
We have not done that, and I have to tell you, this is one of the limitations. Sometimes I do not want to work in a big pharma, that's why I'm working in a small company. But sometimes I wish I was in a big pharma because I could do a lot of these things a lot more easily, that you could try different combinations. There are actually quite a few that mechanistically make sense, but we're just somewhat limited in terms of what we can do. But I can see the logic for it.
Baker, yeah?
I'm just wondering if maybe if I understand this correctly, the vision is to look at strategies where you put a dual payload onto a nanoparticle. I mean, that's not what you're talking about now, but what is your strategy in terms of maybe differentiating PK by using different linkers with multiple molecules?
Yeah, so great question. So that we are actually just starting to explore that because there are lots of reasons why that would make sense. We're starting out saying let's work with approved drugs that we can layer on top of because that's testable. You can show the synergies, you can drive that. But there's clearly a part also that says if we could combine different drugs in the same nanoparticle maybe, you achieve some benefits, not least of which from a regulatory point of view make some of these combinations actually quite attractive. So it's clearly something that I think in terms of next evolutions is going to be on the horizon.
Okay, wonderful. Thank you so much.
All right, thank you.